Hello and welcome, everybody. It's so good to see you all here. Thank you for joining us today for our first Grouper Moon live stream of 2024. My name's Todd Bohannon. I'm an educator from Seattle, Washington, and I've been working with Reef and the DOE on the Grouper Moon project for the past 13 years. I feel very fortunate to be able to be here and participating in this wonderful statement. Okay, good. I'm glad you guys can hear me. Great. And remember, use your chat that's on the right-hand side of your screen. If you have anything to talk, if you want to communicate with me throughout this live stream, that's a good place to do it. So a little bit about me. I'm a teacher. I teach in Seattle. As I said, I've been teaching for about 25 years. And for the last 13 years, I've been working with DOE and Reef to create lesson plans and educational activities for the Grouper Moon Project. And as you know, the Grouper Moon Project is an incredibly successful conservation project, and we're all really proud of it. So enough about me. I have a bunch of people that I'm going to introduce to you today, a bunch of amazing scientists and researchers that are working on the project. I'm going to have some video clips that we're going to share from you of the spawning that started last night. It's incredible. And then we'll have time for you guys to ask some questions towards the end. All right, so we're going to get started. I'm going to introduce you to, to Dr. Croy McCoy, who is the research manager and CIG lead on the project, and Paul Chin, who's a research officer and works on multiple projects with the DOE, including the Grouper Moon project. So I'm going to turn us over so that I'm going to have you guys scooch over here and get real close. Okay, I'm going to cut myself out so we can see you guys. So here we have Dr. Croy McCoy and Paul Chin, research officer. Can you guys, maybe starting with, can you talk a little bit about how this project got started?
Well, first of all, I attended Spot Bay primary for you guys that are on there. And a 29 and a half year veteran of the DOE working on various science projects. And the lead, as Todd mentioned, group of permanent government. The journey with the Nassau Grouper started back in the 90s when I joined the DOE after college in Grand Cayman. And back then, it was still being fished, and they were fished beyond the capacity that fishermen were actually coming and asking us what's going on. And then back
What do you mean they were fishing beyond capacity? What does that mean?
The extraction rate was far beyond.
So they were fishing, they were taking too many fish out.
Exactly.
Okay.
And there was no, season to take the groupers.
So there was no regulation.
No regulation whatsoever, and that led to the grouper hole, as we call them, becoming non-existent or unproductive, I guess you could call it.
So not enough fish.
Not enough fish. Yeah.
Okay.
Then in 2003, the two Cayman Brackers, Jimmy Robertson from Spot Bay and Jim Tatum from the Water in Place area, discovered aggregation on Little Cayman on the west end. And that's where the work sort of started, because one of the reef members were on Little Cayman. We needed help to wrap our heads around how we go about doing a stock assessment, how much is there, and all that we needed to do.
Why is it important to know how, I'm sorry to interrupt you, Croy.
Okay.
Why is it important to know how many fish are out there?
Well, to manage any species of fish, you have to do the biology and ecology of the animal. And then it first starts with the stock assessment, we call it, where you get actual numbers of what's there and how to manage it, really.
So you need to know how many are there to help manage them.
And that collaboration started back then, and it's existed since. So we have a huge team of brains, as I call it. With it takes a lot to get the work done and put everything together to develop a management plan that we are actually seeing the fruits of it now, where out of season you can actually go and catch an Nassau grouper. And that took a lot of work and over a decade of work. And even over a decade, we did not see much returns. And, everything was slow in nature. You don't wreck a system and expect it to bounce back overnight.
Right, so you can't fish all of them out, protect them for 1 year, and expect all those fish to show back out, to replenish themselves. It's going to take, how long did it take for them to start to replenish themselves after the regulations were put in place?
We almost saw, it was almost a decade, but once it got going again, it just, started seeing more fish showing up and stuff. So these systems have elasticity in it.
What does that mean?
I mean, the ability to bounce back, but it takes a certain number of fish to build it to that. So it
seems like there's sort of a critical mass, or like it has to get to a certain number, and then you really start to see the numbers increase.
Yeah, and I call the fish spawning aggregation the maternity wards of our ocean, because any mass spawners, we call them, that release eggs into the water column that go planktonic or oceanic for several weeks, like Nassau grouper, most fish between 35 and 42 days. And I always use the analogy that if you take the mothers, go into the maternity ward, you're not going to have a population.
Exactly.
So that's the
so the idea is to stop fishing while they're actually reproducing during that time of spawning, so that they can spawn and replenish themselves. But then other times a year they can be fished with regulations.
Yeah, and there's this belief that the Nassau group came from different islands, Picklebank and all these other places, and we had to prove that was, if it was happening or it was not happening, and that took a couple of years. We tagged the fish with acoustic tags and put hydrophones around, listen devices, what they are.
And you guys might remember that from watching the Changing Seas documentary. There was a section where they were catching grouper and little tags inside their bellies, and then re-releasing them, and then putting hydrophones, which are like microphones, under the water, and those were picking up those acoustic tags as they moved around the island, right?
Yeah, it gave us movement, told us where they lived, their home range, because they're territorial normally throughout the year except for during spawning time. So, we actually understood what was going on in their world, in the sense of where they fed, where did they go, which rock they lived under, because these groupers actually, where they settle out as juveniles, they live their entire lives there, except for during spawning. I mean, they forage, to feed and stuff, but during spawn, then they go to the west end of the island, to have their babies.
And as if any of you read the Grouper Moon book by Cindy Shaw, or watched the documentary Changing Seas, you know that for most of the year, Nassau live by themselves. They don't hang out with other Nassau. They're territorial. They have their one spot that they hang out at basically their whole lives. But then during the first full moon in the winter, all of them will get together at this, at the Grouper Hole, right, which is on the west end of Little Cayman, and thousands of them hang out for a few days during this spawning event.
Yeah, and when after what everyone knows from the documentary, that the population was fished down to about 1,000, 1,200 fish on the west end of Little Cayman, and that population has since, with all the management plans in place, have risen to 8,000, 10,000 that we have right now, and the BRAC was less than 500, and that's 3,000 or 4,000 fish right now. So we're headed in the right direction, if you want to put it that way, and hopefully it will continue. And 10 years from now, you yourselves will be out there seeing Nassau groupers. It's the largest known population in the whole region, and as Todd mentioned, it's one of probably the most successful fisheries management project in the region and also globally. So we can be proud of that. Every Caymanian can be proud of that fact.
Absolutely. I want to introduce Paul Chin, who's a research assistant,
Yeah, a research officer.
Research officer,
Yeah.
Thank you. Can you tell us a little bit about what your role is at DOE?
Good morning, everyone. I'm Paul Chin. I work with the Department of Environment, and I've been there for 15 years. And I have the privilege of saying that I've been a part of the Grouper Moon project for about 13 years. So I've been on the program for a while, but before that, I was actually sitting in the same seats that you guys are sitting in a few years ago. So if this is a route that you guys are looking to take, by all means, it's not an easy route, but
How did you get there? Like,
Well-
how did it start?
believe it or not, I wasn't originally slated to be a scientist. I wanted to be an engineer, a mechanical engineer, and it took a life changing moment for me while I was on a dive doing some volunteer work for the World Wildlife Federation, where I saw a pregnant seahorse, and after doing some research, I found that the pregnant seahorse is actually male, and the males carry the babies, and exactly. So that was a life changing moment for me, and it's almost like a light bulb went off, and almost immediately changed my degree, and I've been doing marine biology ever since, and I'm thankful for where I'm at. So being with the department, I've been under Croy's supervision the entire time, and he's guided me and helped me to where I'm at today, and I'm very thankful for that.
So if we have some year 4 students out there that are thinking, I want to do that. That's what I want to do. Is there any advice you could give to them or things that they should be thinking about?
My journey with that, I started diving in the Brack in Lower Cayman very young. I started at 9, and it's the only job I've really had is ocean sciences. So the curiosity let me learn more and stuff, and that eventually turned into headed off to university to study marine biology. And then even, once I got, comfortable with that whole arena, it's all I ever knew, really. And then I wanted to further my education, so I kept going, did my master's in marine biology, and then my doctorate degree in marine biology also.
So how much school did you actually go to be where you are now, your roles in DOE?
Well, I spent 4 years, 91 to 95 at University of Tampa, and then I did my master's in University of Wales in Bangor, North Wales.
Okay. Oh, wow.
And then I continued with my PhD there to do my doctorate degree at Bangor University. The former, it was called University of Wales, and it became an independent university. It was a satellite of the main university in Wales.
Okay.
They have several campuses.
I see.
So
My journey started out. I was in high school in Grand Cayman, so I was at John Gray High School.
Woo, John Gray.
John Gray. So we don't have any on here, but prior to that, I was at Red Bay Primary. I don't know if you have any Red Bay Primary on board, but that's where I went. And then I went out to a junior college in Hong Kong where I spent 2 years. And then I did a 5-year program in University of Essex where I obtained a bachelor's and a master's in marine biology.
Amazing.
Yeah.
So can you tell us a little bit, what does the day look like for a scientist or research officer on the Grouper Moon project? How does it start? What are you doing?
Well, it starts on the night before, organizing everything, because the day starts at 8 o'clock on the boat. So
8 o'clock in the morning, you guys are on the boat.
On the water, yeah.
And how many of you are out there on that boat?
usually at least 12,
Okay.
12 to 14 plus crew.
And you take the boat out to the aggregation site, the grouper hole, and then what do you do?
Well, we have different people assigned different tasks. Some are we have this. We're using AI now to actually recognize fish. So we take GoPros and we take video and pictures.
So you guys, I'm sorry to interrupt you. The AI that Croy's talking about is essentially the same that you have on your cell phones. That you hold up your phone, it sees your face, and it unlocks your phone.
Right.
That's this. It's essentially the same type of programming that they're using to create, like a fish Facebook, a database of all of the fish faces because they're all unique.
Right. Like
Okay.
like a fingerprint.
Right.
So each fish has a specific pattern, and the AI engine recognizes those patterns, and then they try to match up the patterns from previous pictures or other pictures that have been uploaded to the program.
Now, what would happen if you were looking at the fish, you saw a fish 1 year and then you saw it again 4 years later? Is it still going to look the same?
Yeah, the patterns is what the AI picks up.
And the patterns stay the same?
Yeah, it's just the same as how we ourselves, our faces, even though human beings, we recognize a face.
Right.
And it don't change much. And
Yeah.
it's the same thing with an also grouper.
Yeah.
So future this year and next year, 10 years from now, the patterns will show up and it will tell you that's the same fish.
That's amazing.
It is, yeah.
So, okay, so you're doing fish faces. What else are you doing out there?
We're doing stereo video. Paul can talk about
So
that.
stereo video. Paul, can you talk a little bit about what stereo video is?
Stereo video, we have a contraption. It's collapsible. It was actually designed by some of the team on some of the reef team. It's collapsible, so it's kind of like how your face works. We have two eyes, which eventually comes together to make one photo, one
Image.
picture, one
Yeah.
image, exactly. So we have two GoPros mounted slightly at an angle, so that way it captures the fish from two different angles and creates one image, and that allows us. Here we go. So here, as I mentioned before, it's collapsible. Once you take this pin out here, it can fold in half, and on either end you have the GoPro.
GoPro.
The GoPro here and the GoPro. And those, even though it looks like straight, they're slightly angled towards each other, and that will allow us to capture the fish into one image, and that allows us to be able to measure the fish as well. So this is a very, very intelligent piece of piece of equipment.
Yeah.
Exactly.
I
Exactly.
can remember the first one we built using that, actually at Hato Bay in the workshop there. We took a base with one of the old high 8 cameras, actually.
Hi.
Yeah,
Yeah.
it's like a cassette, guys. Some of you kids probably won't know what
Yeah, they don't know. They don't know what a video
But yeah.
cassette is.
We put 2 lasers into it, and that's one of the times we figured out that fish don't like the green light, because the minute you shine a green light, they will take off, but the red they don't pay attention to. And we could adjust it. We were adjusting at 25 centimeters. So these two little dots, if you set them at 25 centimeters, 25 feet away, and you adjust it, it doesn't matter how far away it is, it's still going to be 25 meters.
They're parallel, guys.
Yeah.
You remember that from math. Parallel, right? 2 lines that go straight and never cross.
And what we used to do is take video, and once you know that you put that on fish side on, and between those 2 dots on that fish is 25 centimeters, you can extrapolate and see the full length of the fish, and that's how we were doing size classes.
So you're measuring
Yeah.
them using
Lasers,
using
yeah.
lasers that were parallel and
Right.
taking a picture of the side of the fish,
Right.
and then you could from that figure
Right.
out exactly how
Right.
big the fish is.
Then we graduated into this guy here that made
Right.
life a lot easier.
Now, why is it important for us to know the size of all these fish? Why is that part of the data that you're interested?
Well, it serves many purposes, but the main one is to see how healthy the population is in terms of, are we getting what we call recruitment, which is the teenagers coming in to
Or
join
young kids.
or young kids join the
So the new
aggregation,
young
yeah.
ones coming in.
or the big
Okay.
mature ones. Yeah.
And
So the older they are, obviously, the bigger they
larger,
are,
exactly.
right? And the younger they are, the smaller. So that's going to tell you age, right?
Yeah.
Essentially, yes.
we know they recruit to the aggregation to participate around 6 or 7 years, I think. With that, and it also tells us if we measure enough, we get an average, size of the fish on the aggregation. But if we see the population average size starts to move back smaller,
Right.
that's telling us that there's more coming in.
There's more young ones
Ones
coming
coming
in,
in,
right.
and so we want to see more of the
yes.
smaller ones
Yeah,
out
exactly,
there. And
exactly.
is that what you're seeing? Are you?
That's exactly what we've seen.
Wow.
It took about 10 years for us to start to see that pattern show up in the population, which is great. And really, that was a very, I guess you'd call it, aha moment.
Yeah.
That what we're doing is working.
Right.
So, that's very positive news when we saw that.
Awesome.
It took over a decade, as I mentioned earlier.
Yeah, a long time.
Yeah.
You guys, Dr. McCoy, Paul, thank you so much for being here.
It's a pleasure.
And you guys, thank them for being here. Give them a woo.
Thanks, guys.
All right, we're gonna and now
You can go Spot Bay.
Yeah. And now I'm going to introduce to you guys another scientist working on the project. So you guys can hop off there.
Pancakes and eggs awake.
All right. So here we have Dr. Christy Pattengill. Simmons.
Hi everybody.
And so can you tell us what your role is on the Grouper Moon project?
Absolutely, yeah. So I'm one of the directors at Reef Environmental Education Foundation. And so I have been part of the project since the early days, and help in collaboration with some of the other lead scientists and Dr. McCoy and the others from the DOE in helping kind of plan holistically what the objectives of the science, what we want to try and get done in each field season, but also trying to coordinate with education and outreach.
So the objective
broadly.
on any science project, the objective is critical, and to understand that. So what is the objective?
Yeah.
Why are we out here?
The overarching plan is help what's happening, spawning aggregation on all of the Cayman Islands. We focus a lot of our work on this one on Little Cayman, but we also do several trips over to Cayman Brac, and we've worked previously on Grand Cayman to help better understand what is happening each year, change over time. As Paul and Croy were saying, it's been really great to have those aha moments of, the seeing the protections that the Cayman government has put in place to pay off and see that population grow. And so we're doing that through a lot of different ways. We do it, high tech, like with the stereo video camera that Paul was talking about, to just as low tech as looking and watching, documenting behavior, and kind of everything in between. We use a lot of tools in the toolbox and try and just stay, make sure we're keeping track of all the bits.
Right. Okay. So, what you work for Reef, and what is your role?
So I'm a director at Reef, and I work on a lot of the science, the conservation science. Our director of conservation science, Dr. Ali Condelmo, is also part of the project, and we collaboratively bring the work in collaboration with the DOE on Grouper Moon. We're the two primary partners, and then we have our scientists from Scripps Oceanography and Oregon State and other partners as well, to make it really a collaborative project. It really couldn't happen without all of the players.
Yeah, it's a whole village of people to bring this project together. Now, you recently received an award, for a pretty prestigious award. What was that?
Yes, I was inducted into the Women's Diver Hall of Fame.
Woo!
Yeah.
That's amazing. What does that mean? What did you have to do?
Yeah, it's really a big honor. There's just a couple hundred women in the hall, and it was started to really recognize pioneers in the field of diving across a broad spectrum. So for me, my overarching conservation.
All right, we're back.
So my role as part of I've been with Reef over 20 years, and so my contribution and the reason that I was inducted was through my contributions through Reef's conservation work, and the Grouper Moon project is one of those parts. But there's also the hall also recognizes the women in the military and in diving medicine and technical diving, so in training, bringing in women, young girls into the sport of scuba diving.
Amazing. I wonder which of these students is also going to be inducted to the Women's Diving Hall of Fame. I'm sure we've got at least one out there.
I bet.
So we have some footage that I wanted to share that was shot last night, but I want to introduce it first. So you guys know from watching the documentary and doing any of the activities that you've done that these grouper, they spend all year alone on their little home reef site, and then on this first full moon they come together to spawn. And we're out there counting and waiting for that spawning to happen. And when it does happen, well, has it happened yet?
Yes. So we've had spawning for the last 2 nights.
Wow.
The first 2 nights ago was just a little starter, and then last night it was quite a lot.
Okay, so I'm going to share a clip with you guys so that you can see. I'm actually going to share a couple of clips with you so you can see what the spawning looks like.
Yes. So
Sorry. Okay. We were
Yes.
muted.
So-
We're back.
I'll tell you a little bit about what you just saw. So they only spawn right at dusk, right when the sun is setting. So during the day, they do a lot of resting. They do visiting cleaning stations, and
What's
a lot
a cleaning
of
station?
cleaning station is kind of like a car wash for fish. They're actually in known spots, kind of like
Yeah.
your car wash might be on the corner down the street. There are usually set up in sponges or a little outcropping, and there are cleaner animals, little fish mostly, and some shrimps. That's their and wrasse. That's their role, is to come and they pick parasites and dead skin, and they just kind of clean the fish up. And fish use those cleaning stations often regularly, but at the spawning site, they're really important because the fish are spending a lot of energy in being there, and they're all bunched up. They're in a big tight group, so it's really important for those spawning aggregations to have a lot of cleaning stations. So there's a lot of cleaning that happens during the day. And,
So you guys, I will post a video of the cleaning stations on the blog today. So I encourage you all to go to the Grouper Moon Education blog, either at home or in your classroom, and you can see the videos there of the cleaning stations.
Yeah.
Okay, so
that's a really on all coral reefs, but in the spawning aggregation for sure. So a lot of during the day is that, and then as it approaches the evening, on the nights when they're going to spawn, they all end up taking on that black and white. We call it their tuxedos because they get all fancy. They turn on the black
change
and
color.
white. Yeah, that's males and females both will take on this black and white. So that's kind of when you know that spawning is going to happen at any time because they all turn black and white. Whereas before, some were dark, some were light, some were regular color.
Why do they
black
do-
and white? We think it's kind of a behavioral cue. Like, okay, now
it's a
the
communication.
time. Yes.
They're communicating through changing their color.
Yes.
Is that common among fish?
Yes. Yeah.
Really?
Yeah. In nature, there's a lot of different ways that animals can communicate with visual cues,
Right.
and color pattern is definitely one of them. Those fish are not going to use those colors when they're on the reef normally. They're going to be in their regular barred color.
Okay.
But yeah, and then the females, when they're ready to spawn, they'll turn all black and they'll start to rise up, and a bunch of the males will follow after the black and white ones. So what you saw there in that video was a female going up, and usually they kind of go up in bursts, and that burst, there was maybe 30 fish in that one. Sometimes there'll be 3 or 4 or 5. Sometimes there's 50 or 60 fish. And once kind of you get down there, and some will go, and then everybody's going all at once. So us as scientists, mostly our job is to just sit back and watch. We don't want to interfere too much. We're going to interact mostly by watching and filming from the side. But we do have a couple scientists who are in amongst the fish to collect better image data so that we can get better images to better understand kind of individual behaviors, but also to share with the public and with all you students out there.
So we're going to show another clip and I'm actually going to re-show the one that we showed before because I think it froze for some of you. So let's try again and see if the internet's going to be our friend. This is the second clip. This was also taken just last night, you guys. All right, so what were they doing there when they were swirling around like that?
I don't know quite why they do that, but I think
It looks
it's
like a
just kind
dance.
of they're kind of swirling up, probably kind of as they're swimming up. It's easier than just going straight up,
Right.
probably to keep them kind of lower. but still staying all together in a group as they go up.
And so when they're doing that, that's when they're releasing their gametes.
Yes,
And the spawning is happening.
They're releasing the gametes, and then the in mid-water, then those little babies are going to float off into the currents and be part of the currents for about 45 days, actually.
So how big are the babies?
Well, the little eggs are almost microscopic. You can see them. Sometimes you can catch in your light. You can kind of see them. Maybe later we could.
I'll post some pictures of them on the blog for you guys to see.
And then pretty quickly they hatch, and they're tiny little guys.
Yeah.
And then they just float around as plankton basically for 30 to 45 days.
Now, how do don't they just all get eaten if they're just floating around?
Well, that's why so many are spawned, so that hopefully most of them do get eaten or just drift away, but enough of them get caught in currents and eddies. We know from some of our drifter studies, maybe you can share some of the drifter maps later, that show that the water, when on the nights when they're spawning, the water kind of does these fun little currents of eddies that keep the babies close to the island. So that
you guys might remember that from the documentary, right?
So they stay pretty close to the island, and then eventually they drift out a little, and the currents bring them back to the islands by the time 30 to 45 days, when they're ready to settle. What settling means, they come out of the plankton and they actually join the reef. They'll come down into the seagrass and the mangroves and the little rocky reefs that are in shallow water, and that's where they'll spend then a couple of years kind of hiding out and getting big enough so they can go out on the reef.
All right. I have one more video clip that I want to share with you guys that gives you kind of an idea of just how many fish are out there, because it's pretty incredible. You can see some of the researchers out there counting and collecting data. And there's the fish. Now, how big are those fish? Well, I mean, to see it on the screen is one thing, but to experience it in real life is very different.
Some of the really big ones, can be...
There you go.
Can be really big.
What, 3 feet?
Yeah. And then the little babies, they come to the site when they're about that big, like the teenagers.
Yeah. And so how heavy, like what would one of those big moms
I don't
weigh?
know. What's a big mom? What's a big fish? Big Nassau? 30 pounds.
30 pounds.
30
okay.
pounds, yeah.
Between 20, I mean, they're big.
Yeah.
They're big fish.
They are big fish.
Wonderful. So we have a question here from one of the classrooms is, where was this video taken?
Yeah, so that is taken at the spawning aggregation on the west end of Little Cayman. So just right offshore, not far from here, straight out, kind of where the island, the edge of the reef kind of comes to a point.
Mm-hmm.
That's generally where spawning aggregations are found. So on Cayman Brac, it's on the east end, same thing, out at the end of where the island ends, out on the east end is where the aggregations usually are. And we think that's because it's easier to find,
Mm-hmm.
kind of like your corner store, your corner market, it's on the corner, so it's easier to find, right? That's why we think, and it's also for currents and distribution, to be able to, that's why we think aggregations are on edges of islands, not in the middle.
And so it's not just the Nassau that will use those spots.
Yeah, that's right.
Other species will.
Yeah, these are really special places, and that's why they're really important to protect, because it's not just Nassau grouper. We've seen over 25 different species of grouper and snapper and the jacks all use these special places as their reproduction grounds. So they're really important places for not just Nassau grouper. A lot of other grouper species right now are out there spawning as well.
Okay.
Yeah.
Christy,
yeah.
thank you so much for
Absolutely.
being here.
Thanks,
All
guys.
right. And can you grab Janelle?
Yes.
Okay. We're gonna bring in a graduate student that is gonna come and share with us about some of the research work that she's doing. While we're waiting for her, I'm gonna show that video again because I think it's amazing. All right. So I have with me Janelle.
Hi.
Would you tell us a little bit about what you do here on this project and who you are?
Yeah, so I'm Janelle. I am one of the graduate students in Scott Hippel's lab, which I don't think.
We've not met Scott Hippel, but he's one of the researchers here from Oregon State University.
Right. And for my graduate work, I look at the impacts of climate change, mainly increasing temperature, on the development and early life history stages of Nassau grouper and more recently also tiger grouper. And so at early life history stages, it's when the fish are spawning and they start to form these fertilized eggs that turn into little baby grouper
Right.
or larvae.
Okay. So you're specifically looking at how climate change is impacting the aggregation.
Yeah.
How do you study that? What are you doing?
So in past years, a lot of the divers would physically collect the eggs that are coming out in
So
those
they'll
videos
go
that you've seen.
during that spawning. So you saw those spawning bursts in the clips that I sent you. When those happen, several of the researchers will have Ziploc baggies. That's our fancy scientific tool. And they'll swim through it and they'll collect samples of the eggs.
Yep.
Now, is that going to impact the population by taking that sample?
No. So each female produces millions of eggs in her life. So
And there's thousands of females.
Yeah. So it's a very small portion of eggs that we take back. And most likely, not all of those millions actually survive in the real world.
A very, very small, actually less than 1% actually make
Right.
it.
So the amount of eggs we're taking really don't affect the population at all. Really.
And so what do you do with those eggs?
So we bring them on the boat, bring them back to the lab, and I have bins that I will for about a week. So we raise these eggs.
Oh,
just-
we dropped out, but I think we're back now. Okay.
Okay. So we'll raise these eggs in 4 different temperature treatments. The first 2 are current variability, and
What does that mean, current variability?
It's what we would expect. So the first temperature is 25 Celsius.
So why are you testing temperature? Why is that important?
So we can see climate impacts essentially.
So as the ocean warms, which it is doing,
Yeah.
you're seeing how that impacts the eggs?
Yes. How it impacts their development and their survival rates.
Okay.
So are all the eggs going to survive at 31 degrees Celsius compared to 25 degrees Celsius type of thing?
And what is the temperature out there now?
Right now it's about 27, like 27.2 I think is around a typical temperature. In the past 20 years it's been as low as 25, but now we're seeing closer to 27. And
So it has gone up since the
Yeah.
beginning of this project.
And in the future, we're expecting it to go even higher to 29 and then eventually 31 degrees Celsius.
And what is the impact of that increasing temperature? Like when you so I'm assuming you have some of them in a tank that has 29-degree water. And what happens to those eggs as compared to the 27-degree or the 25-degree eggs?
So survival rates are going down, so we're seeing increased mortality and death in the
So
eggs
more
and
die as the
larvae.
temperature goes up.
Right. But not all of them die. Some do really well, some survive for a few extra days, and some don't. And we think potentially there's a maternal input. So some moms are producing eggs that do better than others.
So some eggs are able to survive and some are not. And
Right.
certain moms are producing those eggs that survive and some moms are producing those that do not.
Exactly.
Okay.
And so the idea is that knowing this, we want the population to be as big as possible. So those genes that those moms have, those really good genes that can handle climate change and increasing temperature keep getting passed on. And so the population has its best fighting chance of surviving increasing temperatures.
So the bigger the aggregation, the higher the chance that that aggregation will survive increasing temperatures. Is that what you're saying?
Exactly. So
Which is
all-
amazing for us here on Little Cayman, where we have, maybe 10,000 fish now and growing pretty fast. Right?
So all of the work that Grouper Moon Project is not only helping right now, but even in the future, the next 50, 100 years, we're hoping that, the work pays off.
And so, can you tell us, maybe some of the students out there might be interested in doing what you're doing. What was your pathway to get here? When did you start to think that this was what you wanted to do, and how did you do it?
Yeah, so as a kid, I loved the environment, the ocean, etc., but I didn't know this was a real career. I thought it was just something people did for fun. I had no idea I could do this. And so going to college, I actually studied biology on a pre-med track where I thought I was going to
What
be
does pre-med mean?
Pre-medical, where you become a doctor, a medical doctor.
Okay.
And so that's what I thought I was going to do. I knew I liked science, but I didn't know what type of jobs were
Sure.
out there. And while in college, I found another department at our school called Marine and Environmental Science. And I took just one class, an elective in that department, and realized I really like this stuff, and you can actually study it for a living. And I realized there are professors and researchers and all these people doing that type of work. And so I changed my major like the next semester, realizing it was possible. And I figured I'd try it until maybe it wasn't fun or it just didn't work out.
Just see how it goes.
Yeah, and it worked out. I got an internship in undergrad that allowed me to study in Moorea, French Polynesia. And
French Polynesia.
Yeah.
And what were you doing there?
I did a lot of algae work, actually.
Really?
A lot of, looking at algal traits.
What are we looking at algae for?
Because a lot of the reefs everywhere, not just in Moorea, are getting taken by algae.
Right.
Corals.
So the algae actually will grow on the coral and kill the coral.
Not necessarily. It's just out competing, so it's
Oh,
taking
it's
a lot of
competing
the
with-
resources that
I
corals
see.
need to. Yeah, and so the
Like taking the nutrients that they might need to
Yeah.
grow.
Yeah.
I see.
and so that was
So
a really
how
cool
did you
project.
study that? What were you doing? Were you out in the water, like collecting
Yeah,
algae?
so we were collecting algae, measuring a bunch of. So traditionally, algae is categorized into different groups that aren't always the most useful. So
Okay, 1 second. I'm going to
we're
introduce you. CIS has to leave really quick, and I just want to answer your question. CIS's question from Axel and Agnes. They'd like to know, how do the grouper know when to spawn, and how do they know it's the full moon? And I'm going to bring in Dr. Croy McCoy to come in and help us answer that question. Croy, could you join us? For a moment. Thank you so much. There we go. Okay, and we're back. So the question was, how do they know it's the full moon? How do they know where to go?
Well, for one, you start with full moon.
Okay.
It's something we still trying to understand, but we know the moon phases influence animal behavior, whether it's in the ocean or whether it's
Right.
above.
And the moon actually affects the currents,
It
right?
affects the currents, and they cue in on these factors that leads them to try and nail down how they do it. We're still trying to figure that one
Right.
out. But we know there's certain cues with currents, with moonlight. There's
So even,
tides
even
and
with
stuff
being out
that-
here 20 years and studying this, there's still a lot we don't know, right?
exactly.
Okay.
Yeah. We're still plodding on, trying to figure this all out ourselves. But Mother Nature, these animals been doing this for millions of years.
Yeah, way before
And
we got here.
Way before we got here, and they just continues on. It's like sort of you could almost parallel it with how birds find their way around from
Sure.
flying through, stopping off here to feed, and then going on to South America. It's a phenomenon that developed over millions of years of how they do that, and the how they know where
Where to go?
through the tracking that we did before with these animals. We know a male actually will leave the aggregation, a mature male, and go around carousing other teenagers, if you want to call them that have no idea where to go.
Yeah, they're like, hey,
And-
follow me.
Yeah, and it's called the migration when you see like 10, 20 of them coming along the edge of the shelf,
So they're migrating.
migrating back to the
So
aggregation.
the olders, the adults are literally like teaching the youngers
Teaching
what to do.
Yeah, and it just gets continues that process in the population over and over. So makes it work because the males are teaching, as you say, the teenagers, if
Yeah.
you want to call them that, where the site is. And we even had data where showing us that some of these males would even go to the old grouper hole at the east end of Lower Cayman and hang on there and realize that there's
Nothing happening.
nothing happening. They'll go right back to the west end.
So, and sometimes I think when you had the acoustic trackers, which I think are still there, that weren't we seeing some of the fish would actually swim around the island multiple times before they
Yeah.
would find the spot?
There was totally, but we think that was actually again, doing the migration of
Okay.
them carousing.
So they were
others.
actually, so at that time they're trying to get more to come around.
Yeah, and there's a lot of courtship behavior that goes on.
What
too.
does that mean, courtship behavior?
Meaning, the males will like nudge the pregnant females, and
Right. So certain behaviors that are showing it's
Yeah.
time for us to spawn.
yeah, it's just that behavior that they exhibit. It just seems the color pattern.
Yeah.
They're that you're ready to spawn and
Right.
stuff. The female goes fully black, and the males they go bicolor and such that are cues again, letting each other know when the spawn happens.
Awesome.
And it's the one of the most amazing things. I think the eggs will hydrate, meaning they
They collect water. They fill
Yeah.
with water.
overnight. I mean, they're pregnant but
Because-
the belly is not as big, and then you go back the next day and the belly is huge. They're ready to spawn.
And that means it's about to happen.
Yeah, there's other cues too, and even like we call it, the local name is heron, is scad. These guys show up because they pluck they're like plankton feeders. They pluck the eggs out of the water.
Right.
So whenever you see those show up, and how they know
So,
spawn's
so
gonna
they're
happen tonight.
knowing even before you guys. When
Yeah.
they show up, that's your cue. You're like,
And that's
okay.
one of the cues. Yeah, here is a school of them coming along, and it's going to be spawned that night.
Right.
And that's something again that has been, developed over thousands and millions of years. And,
Nature's amazing.
Yeah, and other fish cue in on that also, and even the we call it ocean yellowtails. I think it's the blue runner.
Okay.
They show up too.
Yeah,
And
yeah.
and the sharks show up. Because they want supper also.
They
And-
want to know about the sharks. Are you seeing sharks out there?
Yeah.
And why? What are? I mean, has that changed over when you first started and there was 1,200 fish out there, and now 10,000? Has there been an increase in sharks? And what are they doing
shark
when they're out there?
presence, sharks look for opportunities, and a lot of time, it's a sign of a healthy ecosystem.
So
When
we
it's
want to see sharks.
Exactly.
Why is that
And-
so important to have these upper level predators?
Well, for one, it's part of how the system works and Mother Nature works.
And keeps that balance.
Keeps that balance. They're apex predators.
Right.
And I recall a couple of years ago, it was over a dozen of them, trying to get
Pick off some of those
pick
fish.
off some. There's some there with pieces, bits
Yeah,
missing.
yeah, we've
You
we've
know.
seen some Nassau that have big chunks out of them, like Lucky, and they're tough, man. Some of them
they're resilient
survive
fish,
that.
yeah. And they still participate in the spawning
Right.
year after year
Right.
with half of their top missing, as long as it's not the spine, they
Yeah.
can still swim, any of their organs. But it's very interesting with sharks, because it's almost like the sharks wait for them to get distracted spawning, because it's a lot of effort to to nail a Nassau grouper.
Because they're fast. I mean,
Yeah.
they're gone. But when they're distracted, when they're spawning, that's the opportunistic
Yeah.
moment for those sharks
And
to
sharks know
dive
that.
in.
They're not paying attention,
Yeah.
and they just go in and take one like that. So,
And do you think, does that have any impact on the population, or is that?
no, I don't.
No.
It's just Mother Nature doing its thing, and that balance in there of sharks are the I guess you call it the cleaners of our ocean.
Right.
Our ocean would not be healthy without sharks. That's why they're so important in this ecosystem. That
And
works
they're
out
they're
there.
picking off the weak and the sick and keeping
Exactly.
and the healthy ones survive, and that's what keeps a healthy population, right? And so seeing more sharks actually is an indication of a healthy reef.
Exactly.
Okay, that's amazing. We have one more question. The question is it hard to maintain the population of groupers? Great question, you guys.
Well, throughout our work, we know we're predominantly self-recruiting, meaning, our babies off the west end
Are all coming from here.
are coming from here.
Right.
And our drifter work with the larval that
That we
Janelle
talked, yeah.
mentioned, and we know there, it's from the old Cayman, this exchange going on for Cayman brack.
And you've done genetic studies too of what's out there to prove that, oh, these eggs are coming from these moms that are from here on Little Cayman.
Yeah, and the sheer fact that I suspected this a long time ago that we are predominantly self-recruiting, because it was mentioned in the small oceanic islands. You have to maintain, we call it a standing stock, which is your population.
Mm-hmm.
And don't depend on upstream to do that. And maybe that's why it took 10 years for us to start to see the population, go back up.
Right.
And the sheer fact that how Mother Nature works is mainly it's self-sustains itself.
So
It's only every 10, 15 years that you see that, we call it a pulse of upstream exit, shift in the oceanic currents.
Okay.
Come this way, and our drifter work tells us that, because a lot of the drifters we release will start those eddies that Christy was talking about, and they loop, but they're coming right back to the islands.
Exactly. And you guys might remember from the documentary, the Changing Seas documentary, that these drifters, they're like a big sock that has a GPS phone at the top that can send a signal to the satellite, and they'll put those drifters in the water on the nights that they spawn, and those drifters will follow the spawn with the current.
Yeah,
Right?
you release them right into the spawn. It's a big sock, maybe 2 feet across and 10 feet long.
Right.
And it's at 25 feet, so it's not affected by winds or wave action.
So it's just the current
Just the
that's really
currents,
driving
and,
it.
It's the strangest thing to see it move against the waves, against the currents, because a lot of them will take off and head towards Jamaica,
Yeah.
looping.
You're like,
and
oh,
come
whale
back.
shark caught that. It's taking it that way. No, that's the current.
Yeah, it's the current. They follow the currents.
That's amazing.
And we've done even work since then with that week-long following the drifters
Right.
and collecting the eggs to
Yeah.
to ground truth and be sure that what we were doing was correct.
Right.
Yes, it was.
It's
correct.
taken a long time to
Yeah.
get all of that data.
They're using an underwater microscope.
Right. Okay, so guys, we have time for some questions. We've had a couple in the chat. I thank you guys so much for it. If you guys have some questions that you'd like to ask, you can unmute yourself and you can ask those questions, or your teacher can put them into the chat and I can read them out for us. And I just want to, while we're waiting for you guys, let's see here. Does Miss Gale's class have a question? Hi there, we see you. Hello.
I think she's speaking.
Oh, but we can't hear you. We just see you. We don't hear you. So maybe you could type in your question into the chat. It's so good to see you guys. I'm so happy to see everyone showing up today. Oh, it looks like we lost them. All right, so let's go back just a second. I want to wrap up with Janelle. So Janelle has been studying the different temperatures and how those impact the eggs, and
Right.
and we see that the bigger the population of Nassau that we have, the better chances they're going to have of surviving climate impacts, a warming ocean. Is there anything else that you're learning from your research about the impacts that climate is having on Nassau?
You already know this, but Nassau grouper is a critically endangered species. So climate wasn't the first thing we were looking at when trying to understand the population, how to help it grow, and that type of thing. So a lot of this work is more recent. It was more so trying to figure out how the population can get bigger and larger after a lot of the overfishing and overharvest of the species. And so looking at climate now is just a whole nother world that we're just getting into and understanding its impacts. And
Right.
so I'm really excited to get even more info out of that. Oh, it looks like Miss Gale's.
Okay, Miss Gale's class is back. On TV. Well, TV, you're on the live stream with us. Okay, so keep going.
Let's see. So there aren't that many answers right now. That we're still figuring it out. We're still learning more and more every day. And
So that's great information for you guys, because we're going to need some of you to become scientists and join Janelle and the rest of the DOE and Grouper Moon team to study how we can help protect the reef from climate change and a warming ocean. Now, if anyone has a question and they want to type it into the chat, now is a great time to do that. We have just about 2 more minutes before we're going to go. Okay, so Miss Gale says, approximately how many grouper are in the Cayman Islands now? Well, I'm going to bring Dr. McCoy back over here really quick, who's going to answer that question for you. Excellent question.
The numbers we're getting that we haven't fully assessed this year, but we know from last year it's between 8 and 10,000 in the Lower Cayman, and the BRAC is around 3 to 4,000. And
From 500 or a couple hundred.
yeah.
Amazing.
But this one from 1,000, 1,200 fish to 8, 10,000.
Yeah.
And BRAC from less than 500 to 3 or 4,000.
It just gives me the biggest grin on my face every time you say it.
And the one in Grand is hard to assess because it's exposed, it's very deep.
Right.
And
It's deeper than here.
yeah,
I see. So harder to study.
harder to study. You have to do tech diving to really do that.
You have to have like a rebreather. And,
Yeah.
It's
But data that the little data we have on it from a few years ago, is maybe 100 fish or so.
Okay.
But we know the population is increasing because there's a lot of the teenagers, if you want to call them, they big.
so you're seeing more teenagers
Yeah.
out there on Grant.
on Grand, yeah.
That's amazing.
And we're hoping next year to have an ROV.
What's an ROV?
It's
like
like a little camera
Yeah.
with a motor on it.
Yeah.
and tethered.
Like a remote control.
Yeah, it's
Remote operated vehicle.
Remote operated vehicle.
But
Thank
it has a camera on it and it's tethered to like a computer up at the top.
Okay.
And we've tried it over the years, but the trouble we always had with it, with the current was so strong, even with 100 meters of line on it, the bend into it did not allow it, and the motors were not powerful
Yeah.
enough to keep up in the current. So we got to sort of get a commercial grade one to have that capacity, and we're hoping for in the next year.
That's exciting. So next year you're going to want to tune in for that. So we're saying maybe about 10,000 here, three or four over on BRAC,
Yeah.
several hundred on Grand.
Yeah, so if you do the numbers on that, it's about 15,000, but that's not counting the juveniles that don't.
That don't go out. That's just the adults, the ones that we
it's,
can't.
It's,
Yeah.
it's the reproductive population that
I see.
gathers at these sites.
So there's actually more than that.
There's actually more.
Wow.
So if you put that together and just take a guesstimate, there's
Yeah.
at least another, it's a 50-50.
Yeah.
Half of the reproductive population, then half is still out there.
Wow. So it might
so-
actually even be double that number. Wonderful. All right, you guys, that's all the time that we have for today. Thank you, Dr. McCoy. Thank you, Janelle, for joining us. And thank you, Miss Gale's class, for your wonderful question. There was one other question. Approximately how many eggs does each grouper lay? And I can field that one. It depends on how old the Nassau is, on how big and old the female is. The bigger they are, the more that the more gametes they release. And I think the biggest ones will release several million eggs each year when they go out to the grouper site. Is that correct?
Yeah, and groupers are not like humans. They always produce no matter what.
Right.
So there's not a time frame as they get old that they stop.
Right. Until the end. Wow.
So that's very important. The
Protect.
females
Yeah.
and the large males to
Yeah.
have that capacity to
Exactly.
keep reproducing. Because we've proven with science that we do here that the larger females produce better quality eggs. And
Right.
so as far as
So
survivorship,
we want to, we really want to protect them.
Yeah, that's why we have in the open season a slot size. We don't.
So that means like when you can fish, there's certain sizes of fish that you can fish, and you can't catch them outside of those sizes.
No.
So the really big ones, you're saying no,
no, and
because those are the most important for reproduction.
Yeah, and then you have the smaller size classes. You don't want to take the young and you don't want to take the old. You take the ones in the middle, which is 16 to 24 inches.
Right.
I guess you'd call them the teenagers.
The teenagers.
Yeah.
Yeah. So take some of those. All right, wonderful question. I want to thank you all for being here. It was great to see you. Remember to check out the Grouper Moon Education blog. I'm posting videos, behind the scene photographs and interviews there every day. And you guys are going to want to be sure to join us on Thursday at the same time, 10:30. We're going to go for a dive. We're going to go underwater. We're going to take you underwater from your classroom, and you're going to see one of the famous dive sites out here on Little Cayman. Thanks again for everyone being out here. Thank you to all the classrooms that showed up. I want to thank everyone from DOE and Reef for all of their help, and we'll see you next time. Go Grouper Moon. Bye, everybody. Bye. Yeah. All right. Till Thursday. Bye, you guys.